Hello students, welcome to Lab 9. We're going to be talking about enzymes. This is going to be part of your lab, your next lab report. So this is also going to be your, what's called the common assignment, or the signature assignment. So overall, this is going to be just the overall grade for the common assignment and the lab report. Okay. Starting off, the introductions. Every chemical reaction in the cell requires a catalyst, some type of enzyme that speeds up reactions by lowering the activation energy for that reaction. It doesn't change the actual energy, it changes the way that the reactions happen. Activation energy is a barrier that must be lowered in order for the reaction to take place. This may involve bringing two molecules together, breaking a chemical bond, or making it easier for the event for that reaction to occur. Because enzymes are not truly part of that chemical reaction, the enzyme doesn't break apart with those substrates. It's not consumed during the reaction. Therefore, it can be used many times over. Chemical reactions can fall into two different categories. You have exergonic reactions that release energy and endergonic reactions that take in energy. Exergonic reactions usually have some type of heat giving off, and endergonic reactions usually feel kind of cool to the touch. Okay. In a catalytic cycle of an enzyme, you have some type of substrate that comes in, binds to that protein or the enzyme fixes it, an induced fit, and then the products are released from the enzyme. And then that active site is ready for the next protein or the substrate to come in. In our experiment, we'll be working with an enzyme known as catalase. And catalase is normally found in eukaryotic cells such as plants and animals. It can also be found in some bacteria cells, but it will convert hydrogen peroxide into oxygen and water. So hydrogen peroxide is H2O2, and catalase basically cleaves that extra oxygen from the water molecule. So if you've ever gotten a cut or something on your skin and you bring in hydrogen peroxide, it will usually bubble up. That oxygen is what's making those bubbles. Now, you don't want to drink it because it can cause other problems inside of you. But if you just leave it on your skin, it'll usually cause bubbles to form up. Okay. Most enzymes are highly specific. They're only able to interact with one substrate. So in this case, catalase only binds with peroxide. Okay, so single cell must contain thousands of different types of enzymes in order to catalyze all of those thousands of types of chemical reactions. So in our case, in our experiment, what we would be doing is taking potatoes that have catalase in it and using that with hydrogen peroxide. So in this experiment, the specificity of catalase will be tested. So what we would do in one of the experiments is we would take test tube 1, we'd put hydrogen peroxide and catalase in it, and then test tube 2, we'd put water and hydrogen peroxide in it. And then we put test tube 3 with catalase and sucrose in it. In which test tubes would you not expect a reaction to occur? And then you write down your answer, and then you say why. In this case, I would expect water not to have a reaction, because as we've seen before, water is a negative control because it doesn't have any chemical reactants in it in order to produce any type of reaction, right? Water is usually a universal negative control. Now, that being said, hydrogen peroxide is really close to water in the form of chemical relationship, right? H2O2 and H2O is really close, right? So that's also something that you can say they're not going to react because they're really close in chemical reactionness or chemical relationship. Okay. And then you can say the effect of temperature on enzyme activity. In this experiment, the effect of temperature can cause a difference in catalase activity. So you'd still have catalase and hydrogen peroxide, but this case, the catalase is inside the potato juice, and you would set the potato juice at near freezing, right? You would set the potato juice in a cup of probably ice water, and then you would bring in a little bit of hydrogen peroxide, and you would see if there would be bubbling, okay? And you would test to see how high the bubbling got depending on what temperature it is. So you'd see it at test tube 1 near freezing, test tube 2 at room temperature, test tube 3, this would be at water bath 37 degrees. You'd probably see it at probably hot water, tap water, warm water coming out of the tap, and then boiling catalase, so boiling water. Okay. In which tubes do you not expect a reaction to occur? Well, that depends, right? So catalase at 0 degrees Celsius. Do you expect a whole lot of reaction to occur at that point? I'm going to say probably not a whole lot of reaction, because catalase is not going to react a whole lot if it's going to be slowed down. Okay? Catalase and many types of enzymes speed up as it gets higher in temperature. Any type of metabolic reactions speed up as it gets higher in temperature, to a point. Once it gets to its denaturing point, then it quickly goes back down. Okay, so depending on how hot the boiled catalase is, the boiled catalase could be denatured and it could go right back down to 0. So the catalase could not react to the hydrogen peroxide if it's boiled. And it could be, just starting to boil. It could be boiled for a long period of time. It just depends. So it could be denatured at that point. These are things that you kind of have to think of as you're doing this experiment. How long is the boiled catalase been boiled? How high is the temperature at this point? Okay. Now, the effect of pH on enzyme activity. This is one that you might not be able to do at home. Okay? Because you probably won't be able to get too much of these types of materials. You may be able to get hydrogen chloride. You may be able to get peroxide. But you may not be able to get the pH meters. Okay, you may not be able to get the pH strips to see what the pH is before you put the hydrogen peroxide in. So, if you want to do this one, you can, but it's not something that you have to do. Okay, you can choose one of the other ones. So, in this experiment, the effect of pH on catalase will be tested. So, test tube 1, catalase, hydrogen peroxide, and hydrogen chloride. So this has a pH of about 3. This is very acidic. Test tube 2 has hydrogen peroxide and water, neutral pH. And test tube 3 has catalase, hydrogen peroxide, and sodium hydroxide. That's pH 11. That's basic. In which test tubes do you not expect a reaction to occur? So enzymes have a very specific pH in which they're optimum. Okay, catalase is usually optimum around a neutral pH. So if you change that, it may not work right, especially if it goes acidic. So HCl, hydrogen chloride, may not work right. But if you go basic, it may work some. Okay, liver is a source of catalase. If you want to do liver, you can, but potatoes are probably going to be cheaper. You can choose liver if you want to get liver, but I would say use potatoes. This is where you need to design your own experiment. Okay, so in your lab manual, all right, each group is going to need to do this experiment. Okay, so I'll show you a video of one I did during the summer. Okay, I used the potato, I mashed it into a blender. All right, I did a little bit of water, then I poured it into some test tubes. I didn't do the experiment fully, I'm just showing you how I kind of set it up. So, at the end of this video, I'll show you how I kind of did that. But you'll need hydrogen peroxide, you'll need some type of test tube, or some type of glass, or something to put in the potato juice. All right, and then you'll need to kind of set up this experiment at your house. All right, I know that this may be a little bit difficult, but if one person in your group can do it, I would recommend that you try and figure that out. Okay, it's a very cheap experiment. It's not going to break your budget. All right, you just need one potato, some hydrogen peroxide, and some glasses. All right, but you need to design your own experiment using the instructions for the lab beginning on page 20 of the lab manual part 2. Okay, the common assignment for this course, for this experiment, if you look at the class schedule, the common assignment is going to be due on the 26th. The next thing, this lab report is going to be due the following week, which is November 2nd. The lab report is on page 28. That's the rubric. make sure you keep that on schedule. Sample lab report is on page 32. Just like last time, the lab report will have a grading rubric, and there will be a group evaluation for that. Okay, if you have any questions, please let me know. All right, and there will be a video right after this to show you kind of how it's going to be done. Once again, with this one, you can choose either enzyme specificity or effect on temperature. Please don't choose both, right? And if you want to choose effective pH, make sure you're following the directions on that. You don't have to choose that one, right? It's a little bit more difficult than the other two. There is some review for Lab Practical 2 questions at the end of this PowerPoint if you want to go through those. So make sure that you fill those out. So if you have any questions, let me know and I'll get to that. All right, so catalase is going to be in our potato. In our potato, right? And this is a blended potato. I added maybe a cup of water, and what I did was I peeled this, not this potato, but a potato, right? And then what I did was I cut it and I blended it up in a blender. And so all of this in here is basically starch, plant cells, and a lot of enzymes. And one of those enzymes is catalase. There's a lot of dirt on that potato. Okay. So catalase reacts with hydrogen peroxide and is going to bubble up a lot in these test tubes. And keep in mind that you're not going to require tests. I'm not going to require you to have test tubes. You can use shot glasses. You can use tall glasses that have a funneled top to it. Just use something that has a large enough top so that when it bubbles, it's not going to bubble over. Okay. So what we're going to do is we're going to use the temperature experiment, which is experiment 2 in your lab manual. We're not going to use the pH experiment because that's going to be hard to do in your homes. You're not going to really be able to get hydrogen chloride. You're not going to be able to get all of those chemicals, but you will be able to get different temperatures of water. Okay, so this one I have cold water, just basically water and ice, a little bit of ice. This one I have room temperature water. This one I have warm water, basically just run some hot water in the sink. And this one I have boiling water. So what you're going to want to do, and this is what I did, I have a little syringe here, which I did the exact amount, 5 milliliters of hydrogen peroxide in each of these test tubes. And if, of course, you're not going to have exactly 5 milliliters, but you can use, little measuring spoons. But, try to measure out as exact as you can in each of your glasses. Okay? So try to measure out what you can. All right? Put them into some type of bath, okay, of your solution, of your water, okay? And then you're going to put in your catalase. All right, so let's go ahead and do some of that right now. This may not automatically work. But you see it bubble up? Right? That one actually bubbled over. So that may have been a bit much. But that's what catalase does. So I don't know if I'm going to be able to do the rest of these, because it's probably going to bubble up too much, especially in the hot water. You can even try it in the hot water. I'm not really sure what that's going to do, but so depending on how much hydrogen peroxide you use would depend on how the reaction occurs. So I may have used too much hydrogen peroxide. Also, if you use these glasses, it's probably not going to bubble up this much. I use a relatively small space for it to occur. Also, this is hydrogen peroxide. It is probably going to lighten your clothes. It's probably going to stain other things. So make sure you do it in an area that you don't have a whole lot of things to, destroy. Okay, thank you very much.